Okay, so epigenetics. Now we need to be a little bit careful with epigenetics if you're reading stuff because people have slightly different definitions of epigenetics. So let's stick with the one that the AQA is written in their specification. They say it's a heritable change in, so you can inherit it in gene function, but you do not have any changes to the base sequence of the DNA. So something else must happen to the arrangement of the gene, but certainly not its base sequence. We're going to look at those other things that can happen which change the function of a gene. Okay, then we need to think about there's two areas we need to think about. First of all, transgenerational in red here, epigenetic inheritance. So that means where you get a change in the gene which is passed on from a parent to a child and grandchild and so on. That is a little bit more controversial to what extent that happens, so we're going to leave that till the end. But what certainly isn't controversial here is epigenetic inheritance, which is occurring from cell to cell. So in other words, in the same organism. We get cells are dividing by mitosis all the time, and some of those epigenetic changes are definitely passed on from cell to cell, and that's fairly obvious that must be happening, because let's have a think about stem cells here. So stem cells are there. Okay, so they have the ability to differentiate into different sorts of cells. So we'd have a totipotent stem cell here. That kind of cell can differentiate into any kind of cell in the body. The only ones you really get of those, of course, are in the early embryo, really early. So when it's just, maybe 20 cells or something. After that, they lose that ability to turn into anything. Pluripotent, well, you get them in the embryo, so they can cells which can turn into a variety of different cells by cell division and differentiation. Multipotent, you do get some multipotent stem cells in adults, e.g. in the bone marrow. There are some stem cells which can divide into a different variety of white blood cells with lymphocytes, macrophages, so forth. And finally, then you get unipotent cells which can divide, but only into one type. So let's think about the cells of the basal layer in the sort of skin, the basal epithelia epithelium cells. They can divide, but they can only divide into skin cells. All of these cells, they've all got exactly the same, they've all got the same DNA sequence, same base sequence, but they behave in very different ways, and they just transcribe different genes or different sets of genes. And as we'll see, epigenetic changes are responsible for this specialization and also a loss of function. So an awful lot of genes are silenced as they go through the. So a skin cell can't make hemoglobin, it can't make insulin, it can't do all those sorts of things. It just does make the set of proteins that skin cells have. But it has all of the full set of chromosomes, the full human genome there. So something has certainly happened there. Right, let's look at mechanisms then. Right, the first one, mechanisms in which genes are modified epigenetically. And the first one we're going to look at is acetylation of histones. Now, before we start to explain what that is, I should say the DNA in prokaryotes is just a naked DNA chain. But in eukaryotes, it's arranged in these units called nucleosomes. So this would be one nucleosome. That blue there is a histone protein, and wrapped around that you have a couple of turns of DNA. So these are proteins called histones. There's 2 lots of 4 different proteins to make up one nucleosome. So there's 8 histones in there. So it's called an octamer. And these histones are arranged so you get you get DNA wrapped around each one. And they're all joined together like strings on a bead. Okay, so that is how DNA is found in eukaryotes, and the histones are quite intimately involved in controlling to what extent genes can be expressed, to what extent they can be transcribed. Okay, so histones contain a lot of the amino acid, the basic amino acid, so opposite of acidic, lysine. They've got a lot of lysine residues. Now, a lysine residue is like this. You have, so the R group is like that. Don't worry too much. You don't need to remember that. But it's got a nitrogen on the end with 3 hydrogens and a positive charge on the nitrogen. Right. That can be acetylated. So you transfer an acyl group to that. So I'll draw that here, and you need acetyl-CoA for that, to do that, which is of course involved in the link reaction in glycolysis, you may remember. And when you stick on an acetyl group, or we call it in chemistry an ethanoyl group, the nitrogen loses its positive charge. So there's the acetyl group transferred to it here. Just put it in that yellow bit, and it means that the nitrogen no longer has that positive charge on it. Okay, now why is that important? Right, and that can be deacetylated that way. Okay, why is it important? Well, this is neutral, and this one here is positive. So this positive one is attracted to the negative DNA strand. DNA is negative because it's got loads of phosphate groups in it with negative charge in it. So when it's like that, when it's deacetylated, it's very strongly attracted, and it forms a tight complex. And the genes are effectively silenced there. They can't be transcribed because all the RNA polymerase and stuff can't get in there to transcribe them because it's too tightly bound to the DNA. Once, and this one here, though, you've got a looser association. It's not positively charged. It's neutral now. So you've got a looser association with the DNA. Got a looser association, so that means that it can be transcribed. So it's active. Right, so they've identified enzymes which can do this. So you can look at an enzyme here which I've doing in red, one enzyme, let's call it enzyme 1. It can deacetylate it. And you've got another different enzyme, enzyme 2, which can acetylate it. And these are targets. People have thought about these as targets potentially for drug therapies, particularly in cancer, when you get genes which are, we'll talk about it in a minute, which are underactive or overactive. If you could modify their behavior, you could stop cells becoming malignant. Okay, so this is acetylation, and that's the first method in which gene activity is epigenetically modified. The second way is by methylation, not of the histones. This time it's methylation of cytosine residues. And okay, now it's not just any old cytosine residues, but you tend to have these regions in DNA called, they're called C, blue, black, sorry. They're called CPG islands. Well, what that means is that P stands for phosphate there, C is cytosine, G is guanine. So you have like lots of many repeats of CG, and you tend to find them in regions of DNA. And these islands are often close to the transcription factor binding sites. They're not found in the coding regions, obviously, because you couldn't code from it because it's just C's and G's. They're found in these regulatory regions, and it's generally high levels of methylation means the gene is off, gene is silenced. Okay, so and then low levels of methylation, obviously the gene is active, right? And the level of methylation can change during the level of acetylation will change as a cell undergoes differentiation in an embryo. So these two mechanisms are very important in controlling cell differentiation. They're probably also pretty important in cancer as well. So let's just have a little think about cancer. Well, in cancer, if you looked at earlier videos, there are two kinds of genes which are pretty important in causing cancer development. Number one, we've got the tumor suppressor genes. Now, if they are off, you've got increased incidence of a cell becoming cancerous. Okay, and the opposite of that, we have the oncogenes, and if they are expressed too much, you have an increase in a cell might become cancerous. Okay, now so tumor suppressor genes, if you think about them, if you remember that quite often people will have you've got two copies of each one, but if you have got one bad copy which doesn't work, then you're at an increased risk of getting cancer. So if you're working with only just one good copy of a tumor suppressor gene, and then by epigenetic changes it becomes switched off, obviously that may push the cell into becoming malignant. Oncogenes is going to be the opposite way around. Okay, so if you have if you're getting overexpression of that because the epigenetic control of it has gone wrong, then you're more likely to develop cancer. And a lot of cancer cells in cancer cells, if you look at tumor cells, they have abnormal methylation patterns for the DNA. And that is definitely a target. People are looking at ways of controlling and intervening the level of methylation as a potential way of stopping cells becoming malignant. Right, before we finish, let's get on to the transgenerational aspect of epigenetics. So this is where transgenerational inheritance. All right, now that is a little bit more controversial. If it actually occurs, or if it does occur, how important it is, because there's two different times, there's two occasions when all the epigenetic imprinting, if you like, is removed. Okay, now that occurs in two times. So the first time it occurs in the cells undergoing meiosis. And of course, they're going to give rise to the gametes. So the gametes will not have any epigenetic imprinting them. In particular, they will become demethylated. So the methylation pattern is completely lost. Now, people have done some studies and found out, and they've suggested certain regions of the chromosomes, particularly like the centromeres, maybe that demethylation may not occur 100%. So perhaps there is a possibility of some of this methylating being passed on from parent to child. The second time in which the cells undergo another demethylation is at fertilization. You get extensive demethylation, and in the male, the histones are all replaced. When the male sperm enters the female egg, it loses all of its histones and it gets replaced with histones from the female cell. Right, so there appears to be ways, these would suggest that you do not get transgenerational inheritance. However, there have been some studies, as I say, which show that maybe the demethylation is not occurring 100%. And also there have been some studies where there's not really strong evidence, but there are some studies where, for example, you get poor nutrition of the mother somehow has an effect on the genes in the grandchild or the child. These studies are kind of quite difficult to prove what's happening, because of course you're going to get lots of confusion by what is a genetic effect, what is an environmental effect, and so on. So it is quite difficult to prove these things. Nevertheless, there is some evidence for transgenerational inheritance by epigenetic methods, but to what extent that occurs is still a little bit unsure of that. And we do have to bear in mind that all the epigenetic imprinting is removed not once, but twice. Okay. But let's not forget the importance of epigenetic inheritance in cell differentiation. It's absolutely crucial there.